Welcome.
Good morning. In 2018, it was reported that 47.6 million Americans suffer from mental illnesses, one of which is schizophrenia. This statistic led me to explore parent of origin effects on contextual and cued conditioned threat learning in type 3 neuregulin 1 transmembrane domain mutant mice. Schizophrenia is a complex psychiatric disorder caused by a combination of genetic and environmental risk factors. One of these genetic risk factors is neuregulin 1, a gene which encodes for a series of cell receptors that influence neural development. Type 3 neuregulin 1 is an isoform of neuregulin 1 and spans the cell membrane in a horseshoe-like fashion. The portion of the cell receptor that is inside the cell is the intracellular domain. It can detach from the cell membrane and enter the nucleus to alter gene expression in a process known as back signaling. This back signaling can be disrupted via a valine to leucine mutation in the transmembrane domain of type 3 neuregulin 1. The valine to leucine mutation was originally identified in an isolated population in Costa Rica. It was also shown to be associated with an increased risk of developing schizophrenia. Among the brain regions implicated in schizophrenia, the amygdala and hippocampus are two. The functions of these brain regions can be tested via the associative or conditioned threat learning paradigm. Using this paradigm, I aim to determine whether the valine to leucine mutation was associated with a behavioral phenotype across wild-type heterozygous and mutant mice with the transmembrane mutation. Additionally, I aim to determine whether this behavioral phenotype was influenced by the parent of origin of the mutation. Parent of origin is the parent of whom the mutation is inherited. In order to determine this, I utilized 5 experimental groups. The first 3, wild-type, mutant, and heterozygous, were used for the determination of the behavioral phenotype. The last 2, mutant mother and mutant father, were used for the determination of the parent of origin effect. All experimental groups were separated into females and males and analyzed separately. This study utilized BL6C57 mice. The first day of my protocol consisted of training in which mice were placed into a neutral context and a neutral tone was played. At the termination of the neutral tone, a 2-second 0.8 milliamp foot shock was administered. It feels like a pinch to the mice. This teaches the mice to associate the neutral context and the neutral tone with the aversive stimulus. 24 hours after the training protocol, mice were placed back into the same neutral context and percent freezing was analyzed. Percent freezing is an analysis of the anxiety levels of the mouse, therefore a measure of how much they learn to associate the context with the aversive stimulus. 24 hours after contextual recall, mice were placed into a novel context and the same tone from the first day was played. Again, percent freezing was analyzed. Figure 3 is a schematic of the training protocol. The first 120 seconds was an exploratory behavior of the new context. Then the 30-second tone played, and for the last 2 seconds of the tone, the foot shock was administered. This exploratory behavior followed by tone and foot shock pairing repeated for a total of 3 times. Figure 4 shows the percent freezing for the female mice when analyzed for a genotypic effect. Figure 4a is the training data. The dashed lines indicate the onset of the tone. The percent freezing increases continually across the time zone, therefore indicating that the anxiety levels of the mice is increasing as well. Figure 4b is one of the analyses of the contextual recall. All 480 seconds of contextual recall are grouped into one time bin and then analyzed it using an ANOVA test. Figure 4c is the second analysis of the contextual recall. Mice were grouped into wild-type, heterozygous, and mutant. The first column of each genotypic group is the naive training period. Naive is the exploratory period, the first 120 seconds of training. The second column is the first 2 minutes of contextual recall. And the last column is the naive cue, the exploratory period during the cue recall. Figure 4D is the cue recall. Again, animals were grouped into wild-type, heterozygous, and mutant. The first column is pre-tone. the second column is tone, and the third column is post-tone. For graphs B through D, individual dots represent individual animals. No significant differences were determined for the female mice when grouped by genotype across all 3 days of protocol. Figure 5 is the percent freezing data for the male mice. There were significant differences between mutant mice, wild type, and heterozygous during both contextual recall analyses. The mutant mice display reduced contextual associative learning when compared to wild type and heterozygous in the first comparison, and reduced contextual associative learning when compared to heterozygous mice in the second comparison. Figure 6 is the parent of origin data for the female mice. It is organized the same way as the first 2 graphs. However, the purple diamonds represent the mutant mother group, whereas the blue squares represent the mutant father group. The mutant father female mice display reduced cued associative learning when compared to mutant mother mice. Figure 7 is the male parent of origin data. However, no significant differences were exhibited across training, contextual recall, or cue recall. My results indicate that the type 3 neuregulin 1 intracellular domain could interact with imprinted genes. Imprinted genes are those which are influenced by their parent of origins. Because the type 3 neuregulin 1 intracellular domain must interact with other genes or proteins in order to alter gene expression, it is probable that they are interacting with transcription factors, which tend to be located near or within imprinted genes. My findings must be considered preliminary due to the small sample sizes, specifically that of the female wild-type mice. Additionally, this study did not analyze baseline anxiety or exploratory behaviors. However, future studies could utilize the elevated plus maze or open field tests to analyze these behaviors. Additionally, further investigation is needed to determine the complexity of the interactions of the type 3 neuregulin 1 intracellular domain. Overall, results indicate that mice homozygous for a mutation in the type 3 neuregulin 1 transmembrane domain display reduced contextual associative learning, whereas female mice with a mutation display reduced cued associative learning, female mutant father mice. Additionally, my results indicate that while type 3 neuregulin 1 back signaling contributes to associative learning, it is not the only function of neuregulin 1 that is required. My functions have implications for genome-wide association studies, as genome databases do not contain information on the parent of origin of genes. However, that would be important for the development of a phenotype, specifically the phenotype associated with psychiatric disorders. I would like to thank my mentors, Drs. David Talmadge and Jennifer Wilking, my research teacher, Mrs. Barbie Frank, and my parents. These are my references. Are there any questions?
Okay, I have a couple questions. Could you define associative threat learning?
So it's a synonym for conditioned threat learning. So
What is threat learning?
It's teaching the mice to associate the context and the tone with the aversive stimulus, so the foot shock. It's learning to associate one stimulus with another.
Okay. So what would be the threat? The threat would be?
The foot shock, so mild discomfort.
Foot shock, okay. So it's associating the sound with the upcoming shock.
Yeah, the context as well. Both the context, so like the mice are placed into a cubicle, so there's a different floor, wallpaper, and scent. So they're associating all those things as well as the tone with the foot shock.
Okay, so I want to just ask you a couple things about the study design. How many mice did you have altogether?
Off the top of my head, I do not know, but I believe it was around 40.
40 mice, and they were like kind of half and half, male and female?
Yes.
Or 40 male, 40 female?
Half and half.
Half and
20.
half. So 20 male, 20 female.
Yeah, about. I
Okay.
can count
And can you tell me why you picked this particular strain of mice was selected?
The lab that I work in typically uses these mice. Additionally, other like postgraduates and graduate students utilize this mice strain. So
Does it have something to do with this particular gene or another reason?
there's an established. My mentor used. I wasn't involved in the breeding of the mice because it was a blind study. So they used homologous recombinations or gene-mediated homologous recombination, and they tend to do that in the C57 BL6 mice.
Okay, and the last question I have has to do with, God, I'm losing my thought here. Hold on a second. Heterozygous versus mutant. Does that mean heterozygous versus homozygous for
Yes.
this gene deletion?
It's not.
Or a gene mutation?
Yes.
It's like a valine to leucine or something?
Yes, in the transmembrane domain.
And what is the, do you know what happens when valine goes to leucine? Like what is the problem?
Oh, so the way that the intracellular domain detaches is that gamma secretase would cleave the cell receptor. It's bidirectional signaling. So after the cell receptor signals with like a adjacent cell, the gamma secretase would cleave the transmembrane domain, and then the intracellular domain would detach. But because the valine changes to a leucine, the gamma secretase is no longer compatible as like a cutting enzyme.
Okay, thank you.
Thank you.
Good work.